All right, good morning, everybody. My name is Bob Bruner. I am the exotic forest pest educator with the Department of Entomology at Purdue University, and today is the third session of our fall semester of the Emerald Ash Borer University in 2025. Today we are hosting John Bonkowski, who is a part of Purdue's Plant and Pest Diagnostic Lab, and he is here today to talk to us a little bit about some of the upcoming invasive pathogens that we may be dealing with in a lot of our plants. John, the floor is yours.
Good morning, everybody. Thank you for having me. As Bob had mentioned, I'm work at the PPDL, the Plant Pest Diagnostic Lab. I'm a plant disease diagnostician there, so I work on all the samples that come into the lab. In particular, if there is a disease problem or even an insect issue, we might be the first step that you might take, if not contacting the IDNR, the Indiana Department of Natural Resources, concerning these issues. So I could be a potential point of contact if you come across any possible invasive problem in the state. So I'm going to be discussing diseases on the horizon. Primarily, most of these are things that we kind of already knew about or were kind of coming into contact with each year, or things that might be slowly spreading towards our way. So to begin with, I just wanted to mention that a lot of the disease problems that we deal with, we're worried about them as they impact woody plants and trees because they can reduce their overall plant vigor. Now, as this relates to commodities or things in the natural environment, this is especially important for an ecosystem standpoint as it affects the overall stand of plants and can affect how the microclimate or the general climate in there it kind of affects that microclimate and can affect the plants and animals that are surrounding those that are actually dying. It generally, some of these issues can contribute to plant stress, so that leads into that plant stunting or that plant vigor reduction. And then it can also make them more susceptible to other issues. So it may not necessarily be the particular disease problem that we're worried about that will kill these trees, while it very well could, but it might lead to other things which are that may attack naturally stressed trees that may kill it later on. So the whole idea is that there is this kind of effect cascade that happens. So it affects the ecosystem. There could be a loss of mass or food source for plants, I'm sorry, for animals, for insects and things like that. And so it affects the overall diversity in those locations. There's gaps in the canopy, and so that can lead to changes to the other plants surrounding them, not necessarily just the animals. And so this also allows for other invasives to move into those areas and changes, like I said, those microclimates. Now, on the other side of things, when it's not just a natural environment, we can talk about the commercial side. There are disease problems that will affect the movement of plant materials. So this is a regulatory action, quarantines and things that prevent us from moving things from one state to the next and be sold across the entire US or even to other countries. Now, this may affect the overall availability of certain plants for trade, in particular ornamental plant species. But since these problems can move around in the plant trade and then can also escape into the natural environment, it can then still get into highly desirable trees or plants in the native landscape as well. And so I'll go through a few examples in the future where we have actually seen this in maybe other states, not necessarily here. So the first disease problem I'm going to be talking about is boxwood blight. Now, this doesn't really affect too much our native plants, but ultimately it has a massive impact on the landscape. So as an invasive disease problem that came from out of the country, it is wiping out a very old standing plant that's been used widely through the US and in Europe. Boxwood blight ends up killing these plants, and it originally was first found in 2011 in the United States, out in Connecticut and in Northeast US, and has since spread throughout most of the eastern, well, the entire East Coast, pretty much, and then also the Midwest. A lot of boxwoods are being planted and grown out west, especially in Oregon, and so we're seeing a lot of disease out there as well. Although it's not as necessarily traumatic to those plants since they're growing them out there, they are being disseminated from nurseries out in Oregon elsewhere too. We don't have a more updated map since 2020, but ultimately, I haven't been able to find information on more states where it's been found, but it's been found in 30 states so far in landscape and retail and nursery locations. This disease problem is caused by the fungus Calanectria pseudonaviculata. Like I had mentioned, it causes a lot of damage to boxwoods, especially even older boxwoods, under the right conditions. It really likes humid, moist conditions. And so when the fungus infects the leaves, which it normally starts off as this kind of leaf spot you see on the left-hand side, over time, it'll lead to defoliation. Now, the big thing about this disease issue, so if you have your own boxwoods, you may know that there's damage that can occur, stems might die, leaves might die, but more often than not, most of the problems you see, the leaves stay attached to the branch. With this disease, boxwood blight, it often leads to defoliation. So that's a big thing that can help separate this disease from others, especially if you're, monitoring your own boxwood plants. As the plants defoliate over time, it stresses these plants and it is putting more energy in trying to put new leaves out, which become infected and fall off again. So it's over time a significant reduction in vigor of the plant. And over time, it will die because it just doesn't have any more energy to try and push out new leaves and produce its own food. Now, if you look on the right-hand side, these are really, really old boxwood plants out in Virginia that eventually succumb to boxwood blight. But what's interesting about this is you can see at the very tippy top of the plants, those leaves are green, and so they're still alive. And that is related to moisture. So there's more airflow. Those leaves dry out faster, and so the disease, which relies on water and rain to kind of move around, is not able to infect those leaves as well because they dry out faster. And so that's a very important point with this disease problem. Once we have more moisture, we have more humidity, we can have more disease. Here's an example of kind of topiary or just late hedged boxwoods that are cut into these little small balls in the landscape where there's small areas of the canopy that are dying back because of boxwood blight. You can see there's individual areas of leaf spot or leaf defoliation. And on the right, we have another example of a larger shrub that is slowly defoliating to the disease. Here's a little bit closer image on those leaf spots. Often they will be kind of black in the middle and then spread, which will lead to a tan center of dead tissue. The leading margin might be kind of a red or yellowish brown color as it spreads, but over time it will keep on going and going and might spread to a larger part of the leaf area, and that leaf will eventually fall off. Now, this can also infect the stems. And so even if it loses leaves, there will be the fungus in the stem and it'll be able to stay there for extended periods of time. You'd have to prune out that branch to try and get rid of it. But ultimately, most of the foliage or a lot of the foliage might be infected and have the fungus anyway. So it doesn't really make it an effective strategy to try and limit the disease. These lesions could potentially girdle the stems. Now, girdling means that it will cut off the vascular system as it spreads around the circumference of the stem. Now, what's different with this compared to other disease problems, though, is that these lesions often are longitudinal. So they kind of go up and down. You need a lot of these lesions on the stem to try and cut off that stem because it doesn't normally go around the outside, it just goes up and down along the length of the stem. So we typically see, once again, those that kind of dieback and death, usually because of the loss of vigor and loss of leaves. The fungus, Calanectria, we also call it cylindrocladium. These are both similar names for the same organism. So depending on how old of a fact sheet you look at, you might see both of these names. Calanectria produces very sticky spores that are white in color, and they create tufts on the affected foliage and stems. So you see these under humid conditions. When it's really dry, you wouldn't necessarily see this kind of pop out. But that is what we're looking for to try and verify the presence of this pathogen and identify it as boxwood blight. Now, the fungus itself, once it's in that tissue, it actually produces what we call microsclerotia. So we have spores that spread, and those are very sticky. But we also have these other structures, which are embedded in the tissue. And they are thick-walled survival structures that can survive for long periods of time, months to years. The problem is, once we have leaves that drop off the plant, we're not necessarily picking up every single leaf that comes into contact with the ground. And so this could actually survive for a long period of time in that landscape where the boxwood was planted. So if you plant a new boxwood, it could potentially create that disease issue all over again and infect the new plant. So we have to kind of be aware of all the plant material that's that exists there. And what's kind of interesting with this, once it drops all these leaves, some nurseries actually use flamethrowers to try and destroy that material. Now, might be in like a gravel situation or an area where, that might not necessarily go up in flames. But ultimately, we can destroy that leaf material, but it's going to be harder to do either to collect it all or get rid of it in a landscape scenario. So I had already mentioned that this disease spreads by water splash or wind-driven rain, but since those spores are very, very sticky, they can move around on contaminated tools. So we have pruning shears, we have loppers, things like that, maybe even hedge trimmers, but I'm not sure if it's going to move around on that. But since it's sticky, it'll hold on to that tool, and once you cut from one plant, you can cut on another, you can spread it to the new location. People and plants can also move them. So when you're working with, if you're in like a nursery situation, if you're wearing clothes and you have a situation where you think you might have boxwood blight, you might want to change, shower, wash hands before going into a new location. So typically we would say, work with all the clean material first, then work with the dirty stuff because then you're going to have to kind of decontaminate, if you will, and clean yourself up. This pathogen also moves in the debris. So leaves, like we had mentioned, plant material, as it falls off the plant, it can survive in that for long periods of time. So it could potentially move in those ways. But since this is a plant that's being grown and sold in nurseries and in garden centers, things like that, it moves around in the US plant trade. So that's where it's moving from really long distances. Um, I will say that pretty much the states that do regulate this disease issue, they often have nursery inspectors that go out and inspect plants annually. And so the IDNR conducts an annual nursery inspection for boxwood blight in particular, as well as many other disease problems in the nurseries. And so typically there's about 100 to 200,000 plants that are inspected, and this is throughout 63 nursery garden locations throughout 28 counties. And so it's very extensive and it takes a lot of time each summer to look for it. Anytime they have disease problem that they suspect is boxwood blight, they'll end up sending it to us for verification. So just to kind of give you some numbers over the last few years, in 2018, there was a really bad year where boxwood blight was disseminated from a few different nurseries throughout multiple states. And we've had positives in retail locations, but we also had a positive sample come from a landscape. The next year, it died down. We only had one positive from a positive location, retail location, but still we had a spread of the pathogen from a landscape situation in Indianapolis. And 2020 wasn't much, but then again, it was also COVID. So a lot of people were, not focused on the plants as much as you'd expect. But in 2021, we had another new positive location in the landscape. So since then, we haven't really had too many come into the lab in terms of boxwood blight positive samples, but this is still a problem that we have to worry about as the disease is running rampant in areas where it's very conducive. We are drier in the summers, and it doesn't seem to be as big of a deal, at least from what we can tell. But ultimately, under the right conditions, it can still spread, still kill plants. So we have to be very cautious about this problem coming into our state. We don't know all the locations where boxwood blight would be here in the state, but we just want to spread information for people to kind of be aware of it. So management. How do you manage this? Once you find the problem, we would recommend digging up any infected plants. And if you have a lot of boxwoods around it, digging up those surrounding directly next to those, because it's easy for this fungus to spread from one location to the next with water splash or even handling the plants, right? So we want to put those plants in a garbage bag and send it to the landfill, or if you can burn it on site, do so, which this is usually going to be more likely in areas where there's lots of plant material that need to be taken care of, like in a nursery, and move these plants around as little as possible. You could potentially excavate the soil and picking up all the leaves, but this is very difficult and impractical, especially in a nursery situation or even in a landscape. So we often recommend just trying to pick a boxwood alternative instead. And there are chemical control options, so multiple fungicides that can be applied to try and slow down the infection. But once it's present in the plant, especially on the stem, you can't kill the fungus. So it cannot be cured. You can prevent new infections, but once it's there, it's kind of always there. So you have to be very, very careful with how you deal with this issue. There is a lot of information about boxwood blight online, and so one of the best locations I would recommend going to is the Boxwood Blight Insight Group. So here, boxwoodhealth.org, the Knowledge Center, which is kind of dead center there at the top, has a lot of different pieces of information, especially about how to manage it, what you're seeing, different plants you can plant in that location, and so on. So I would recommend checking that out if you have an opportunity. The next disease issue we'll be talking about is sudden oak death. And so while I end up saying that this is not just for oaks, there's a lot of different host plants that are affected by this, although sudden oak death is the more commonly used name, or remorum blight. But it is a fairly destructive disease, especially in the right locations. So sudden oak death is caused by a water mold. It's an oomycete. So this is an organism that's kind of like a fungus, but it's not really. It grows very well in water and in very moist conditions. And so that is where we're kind of finding it both in the landscape and in natural areas. Um, Growth and pathogenicity is favored by free water. We have multiple organisms that we kind of worry about in these conditions, downy mildews, pythium, but Phytophthora is the main one that we're going to be talking about. So Phytophthora is a very destructive pathogen. It is a soil and waterborne organism. It produces structures or at least spores that can swim. We call them zoospores. So they actively move in the direction of hosts in water when they can, but they are readily taken up and moved in water. And so they can pass down from one location to the next in the flow of a stream or something like that, or even irrigation water. And they often will overwinter in the soil or in plants with survival structures. Just like the microsclerotia with boxwood blight, we have what's called a cometospore or an oospore that can survive in plant material. This can be fairly problematic in wet conditions, like I already mentioned. So when we have really heavy rain, flooding in particular, very saturated soil, or areas with heavy soil texture, this disease can kind of explode. So in nursery situations, when plants are having overhead irrigation and excessive irrigation, this can thrive and kind of spread very quickly. So Phytophthora produces sporangia, like I mentioned. Here on the left-hand side, there are these structures. They're produced on hyphae or the threads of the organism, and that's growing from infected tissue that's floating in water. Once you have a stream, or if you have kind of the movement of water, it can actually pull these off and it'll flow very readily in the water down wherever the water's going. Up close, you can see here on the right-hand side, those sporangia as they've broken off the hyphae. They're kind of lemon-shaped and they have this kind of, we call it a papule. It's a kind of a nipple-like structure at the very tip. But this is can produce its own zoospores or can flow and directly germinate and infect leaves, roots, even stems of certain plants. Now, remorum blight or sudden oak death has been detected out in California initially, and it has spread very widely in natural areas there. It's also found in some nurseries, which is actually kind of the beginning of trace forwards, where they're disseminating plant material that has been found to be infected with Phytophthora worm, and we have to worry about that in states farther east where they're shipping it. But this disease problem is located kind of restricted in California, Oregon, and Washington, where they're finding it year after year. In the United States, we've had a few occurrences of plants where nursery locations where infected material was found in those nursery areas. But at many of these times, we end up eradicating the material and ensuring that it doesn't take a foothold. But of course, we don't necessarily know that it is, I would say, there to stay or not. With constant surveying and sampling, we ensure that we're not getting positive plant samples, but we still aren't 100% certain that it hasn't spread out of these kind of locations. As you can see, it's also been spread throughout the UK, throughout Europe, and it causes damage to, like I said, a large number of plant species. Though oak, kind of in the name, is one that we're really worried about as it pertains to our natural ecosystem and native forests. So with that known distribution, there was actually a very large trace forward event where plants were sent throughout 18 different states. And so there was a number of different nurseries where we found positive samples with Phytophthora remorum and causing damage in rhododendron in particular. There was a very big recall of that infected material to try and get it out of those nurseries and maybe even to destroy it, because once again, if this spreads, it can cause a lot of damage to our native plants. But what was interesting for us in Indiana is that one of the major retailers had a distribution center here. And so a lot of those plants for the recall were being funneled into a specific distribution center in our state. Hopefully everything was managed well and that it didn't kind of escape, but we can say that we haven't had any natural detections or sorry, detections in natural areas. Everything has only so far been in nurseries, and then we don't necessarily know of anything in a landscape scenario. But the important thing that I'm trying to convey here is that pretty much with every single year, there are trace forwards that we have to deal with. Phytophthora moving from a location that's kind of taking a hold out in California, Oregon, Washington, out west, that is sent elsewhere. Now, if we look at this map, which was produced fairly early on and kind of evaluating the risk of Remorum causing damage throughout the US, it kind of shows that there is relatively low risk of damage throughout most of the country. However, we have some spots of high and moderate risk. And so in Indiana, you can see we have a few areas of moderate risk, especially with our oak forests. So, like I had mentioned, it has a fairly wide host range. It can affect over 100 genera of plants. And so some of the key plants that we worry about are oaks, like as the name suggests. Tan oak out in California has suffered significant damage with cankers and dieback related to this pathogen. But a lot of the plants that move around in the nurseries, including rhododendron, azalea, camellia, viburnum, pieris, kalmia, and lilac, tend to be kind of the dirty species that help move this around, unfortunately. Rhododendron being the biggest one. Some of the symptoms we might see with this sudden oak death or Phytophthora can be dieback. So we have complete dieback of portions of the tree, and this might be associated with an actual stem canker or stem infection. Now, when we have this canker, there's going to be a lot of ooze or sap kind of coming out of the trunk. And if you end up peeling away that bark where the sap's coming from, you end up seeing the kind of infection right below the bark. It's going to be outlined in almost a red brown kind of color. On the right hand side, that's a tree. I believe it's the tan oak, and it actually has a reddish coloration underneath the bark. But you can see that kind of water soaked lesion where Phytophthora has infected the wood, and that is where it's going to be starting to ooze this sap, and it's kind of a bleeding canker on the main trunk. Now, this is going to be only some plants. Not all of them will show these kinds of symptoms. The majority of them will have foliar blight. So we call this blight or dieback, where there is a rapid necrosis of the leaf, and we have small little individual lesions that might expand to kill larger portions of the leaf. On the right-hand side, there's California bay laurel, which is actually one of the species that helps keep remorum alive in natural areas in California. It's not damaged as much as other plants, but one thing to note with both of these photos is that the pathogen moves with water. And so the infection starts where water ends up being held or retained. So on the left-hand side, you can see a lot of the water at the base of the leaf because that's where it's kind of, rolling down to. Same thing with the California bay laurel. At the very tips, all that water is going to be kind of coming down to the point, and that's where it infects the leaf and creates that necrotic area. You should be able to see my cursor. Here is the tip of one leaf also infected, and you can see that the infection is starting on the leaf right below it because they're touching, and that's where the pathogen can then easily move into as the water's being held and collected on the leaf. Once it spreads to the main vein, it can then spread very rapidly and might blight out almost the entire leaf if it gets down almost to the petiole. On the flip side, you can actually infect the stems. So once a stem is infected, it'll have this dieback blight, and it can actually spread into the leaves from there. But ultimately, if it's in the stem, it ends up killing that branch, creating that dieback, like I had mentioned. So these are some of the symptoms we're looking out for, especially on rhododendron. Symptoms may be similar to a lot of the other plant species, but depending on the cultivar, depending on the plant species, some of these symptoms might be a little bit different. So we will have this water soaked lesion. So water soaked being, it looks wetter. It looks like on the left-hand side with the viburnum. It's kind of just darker in appearance, but it all might actually be developing this necrosis behind it. And so these symptoms look very similar, but may not be 100% exactly the same between different species. Like I had mentioned, since it moves around in water, it could be spread through water splash. It could be water go through the irrigation water itself, but it needs this excessive moisture. So a lot of the management strategies we would recommend putting in place when dealing with this is, of course, roguing out infected plants where possible, but ultimately letting it kind of dry out and increasing airflow to reduce potential moisture. Now, of course, since this is moving around the plant trade, we want to make sure that we're not going to be moving infected plants from one location to the next where possible. So we want to quarantine plants that come into new locations to at least prevent it from mixing with new plants. But of course, just make sure that the disease and spreading. So try to make sure you get healthy, clean plants where possible. The next problem is thousand cankers. So this is a disease problem that affects walnut. You can see here that the trees are showing significant dieback or are potentially dead due to this issue. Now, what's different between the two previous ones, where there was just a pathogen involved, this one involves a vector. So we have the walnut twig beetle. It's an insect that will attack walnut trees, and it actually has a fungus that it carries. The fungus is Geosmithia morbida. Once it is introduced into the host, it ends up leading to a disease problem. So we kind of need all three of these things to occur to be able to have thousand cankers disease. So the susceptible host, Juglans nigra, or Juglans, some of the Juglans species, so walnuts, are susceptible to this disease problem. What's interesting here is that the beetles will attack en masse on the tree, and they will cause significant damage by tunneling just underneath the bark. And so you can see some of the galleries that they produce by kind of feeding on the tissue just under the bark, creating these little small tunnels, and they spread the fungus as they go. So the fungus will grow in the tunnels, and then eventually the beetle will feed on the fungus, not necessarily the tree itself, as its form of sustenance. And so it is relying on the fungus for its food, and it wants to spread it farther where possible where it goes. So we need the beetle to cause the damage, but we also need the fungus there to create the disease problem. Since this causes significant damage to the tree over time, the fungus will kill the tree and will lead to it showing significant decline and limb dieback and loss. So the beetle causes a lot of damage, but the fungus does a lot more. So we have all these galleries, but here's a canker that develops at that little spot. So if you are attacked over 1,000 times, you end up having 1,000 of these spots that continue to spread and girdle those branches, which eventually will lead to the dieback, like I had mentioned. These little beetles, they're very, very tiny. On the top left, you can see their size in reference to a penny. So you can't really easily see them with the naked eye, or if you can, it's only very, very small, so you can't see any defining features. So you need a microscope to be able to determine that. Here are some wood from a walnut that was infested with the walnut twig beetle, and they collected them, kind of let them escape in like a bag or a net. And they collected them, put them in a vial of alcohol, and you can see all of them just completely filling up that vial. Almost looks kind of like a liquid with how many there are. It's pretty gross when you think about it. And you can see here on the left-hand side, there's 23,040 beetles that came from just those two logs. So once we have an infested and infected tree, here's the dieback we might see. One thing to note is that after there is the initial attack, it may take a long period of time for symptoms to develop. And this is very common for a lot of mature trees. So we have damage to the stems, we have damage to the limbs. Over time, once those cankers start spreading, it will eventually lead to general decline and then death of the branches. This is just kind of a progression here from June of 2008 to 2009, where a significant dieback was observed. And eventually almost near death of the tree that was affected. Now, these problems are also more severe under adverse conditions. So we have rapid decline when there is drought stress or there's lack of water, because ultimately these cankers are affecting the ability of the plant to transmit photosynthates, carbohydrates, their food, and water. And so we end up having a lack of water getting to where it needs to go. Therefore, it's getting stressed and slowly dying. Now, those photos were actually in California or Colorado, out west. But here is a map of all the surveys that were conducted since 2013 up to 2020. There wasn't a whole lot observed where there was walnut twig beetle and Geosmithia morbida. You need both to have the disease problem. And so if you don't have both, we're not going to call it a positive. We don't have TCD or thousand cankers. So there's very few instances where we've actually found it. Now, we have found it in Tennessee, we've found it in Indiana even, and out in Northeast. But after those individual situations, there wasn't really much that happened. So most of what we've been seeing TCD in Western states, it hasn't really been a big problem out here in East, but we still keep an eye out for it as we are having dying or declining walnuts because we are concerned that it could affect a important timber species in our native environments. One thing to note is that there are at least 18 other beetles that can spread Geosmithia morbida, the fungus. And so, since we don't have a beetle that's aggressive and attacking these trees, it's not causing significant stress. There's not tons of cankers being developed. We're not having the dieback we would expect. So, since we don't have walnut twig beetle, we don't end up having significant disease spread or damage. Like I had mentioned, some of the disease plants in Tennessee that were found were in landscape situations or um, out in native environments, and they're not significantly stressed. And so some of these trees that are in natural areas, they end up showing maybe some disease symptoms, but then they recover. Where if they're significantly stressed all the time, it might progress because there's multiple compounding factors that lead to that tree dying. So we're not entirely sure if this is going to be a big problem or not. So far, we think it's not, but we still keep an eye out for this problem altogether. So what can you do? You want to keep your tree healthy in landscape, especially try to keep it watered and fertilized if possible. Mulch around the base of the tree to make sure that there's less drought stress potential in a given summer. You want to avoid damaging the trunk or the roots of the tree that might affect water conducting, elements, and so then leading to this artificial drought stress problem. You want to thin out dead and dying trees because, of course, with these beetles, they're attacking the trunk. They're inside the wood. If it's something that's dying, get rid of it. You're going to get rid of a lot of the wood boring, bark beetles, and things like that. And so you need to chip the wood or burn it to try and get rid of anything that's inside of those. And then, of course, don't move firewood. If don't try to bring anything from out west out to here, or try to just try to buy it locally, so you're not potentially transmitting or sorry, transferring and moving those insects. The next disease issue is laurel wilt. This is similar to TCD in that there is a beetle that spreads a fungus and that causes a disease problem in the plants. Laurel wilt, as the name refers to, it affects laurel trees, so Lauraceae trees, and there's a large number of plant species. We've already seen a lot of damage in the U.S. At first, it was on red bay trees out in the East Coast, and it has devastated the natural population of those trees. Pretty much the only ones that are standing still in some of those locations, like in Florida and Georgia, are those that are naturally resistant. And so that's due to the diversity within the red bay species. And so they're not being attacked by the beetle, or they're not causing as much damage. Sorry, they're not as susceptible to the fungus as other red bays are. So we often see it also on sassafras, and there's actually a big problem of it on guacamole plants, avocados. We grow avocados for guacamole out in Florida, so that's actually in production. The fungus is called Rafflesia loricola. There's a new name, Tarrantonia, but I haven't updated the slide, so I'm going by my preference, the older name. But that is the fungus that's being spread by the red bay ambrosia beetle, Xylabrus liberatus. So what ends up happening, just like with TCD, we have these bark beetles that attack the tree. They infest it. They will spread the fungus, and over time, it will infect the tree and create symptoms, which will lead to wilting and decline. What you don't necessarily see with TCD is that these beetles will bore into the wood and they actually produce toothpicks that kind of push out from the stem. And they will stay there as long as there's not significant wind or even rainfall. And so that is one of the big signs that we can use to show that there is a wood-boring beetle present is the sawdust that they're producing being pushed out of the main stem. On the top right, we have a fungal culture of Rafflesia loricola, and that is what I'm trying to isolate to identify what's present from an infected tree. And bottom right is the beetle itself that spreads it around. Now, these are really tiny beetles as well, and so you can see here in reference to someone's finger, and which I think might have another example in a minute, but that is a very, very small hole. So these are tiny beetles, once again, so it's going to be difficult to just identify it on site. But once they get in there, they're spreading the fungus, and you can see these dark lines. This is vascular discoloration. So it's the vascular tissue inside the wood. This is where it's conducting water and going up and down the tree. What happens is the fungus gets in there, and the tree itself is thinking that it's invaded, which it is, and it is trying to block off the spread of this fungus. And what ends up happening is you have this discoloration forming, and there's water being blocked in these locations, and that leads to wilt of the plant. With significant infection by the fungus, you have lots and lots and lots of these very thin streaks that develop. And on the right-hand side, you can see how it's going up and down in the tree. Now, this is a systemic infection, so it's going to keep on spreading through the whole system, the whole plant, and eventually lead to wilting of the plant, like we saw a second ago. Now, like I had mentioned, hosts are in the Laraceae family, but spicebush, sassafras, that's what we would find here in Indiana, and that's what we would have to be a concern about. But avocados are also affected. So there was actually a campaign of save the guac when it comes to laurel wilt out in Florida, as there is a significant avocado production in South Florida. They're doing a lot to try and deal with this issue, but apparently some of the trees, once they get infested and infected, they just become a beetle factory. And so early detection is very important for them out there. For us, we haven't found it in Indiana just yet. However, right here in Kentucky, there are a number of locations where it's been found over the last few years. And while we haven't had any samples yet that have been positive, we are still kind of aware that it just is easy to jump that line. It doesn't exist anything beyond what's on the map. And so laurel wilt is likely to spread into Indiana from farther south. The next disease problem we'll be talking about is beech leaf disease. This will be the last one. This problem is specific to beeches, and it affects all beech species, to our knowledge, not just native ones. But we are more concerned about spread into native beeches because it can start as a slow kind of damage to the leaves, but over a long period of time lead to decline of both saplings as well as mature trees. Here is that kind of more classic symptom we've been seeing with this issue, which is kind of veinal banding or interveinal banding on the foliage, and it's not uniform across the leaf. There's only individual sections where it spreads, and then some rugosity of the leaf. So I'll show you some more symptoms in a minute. So here is a map of the current distribution of beech leaf disease. It is primarily starting around Ohio because that's where it was originally found in 2012. You can see that little tan spot where it was located, and has since spread throughout the east New England area. And in 2024, it moved into Maine, but we've also observed it going into Canada. We have some collaborators and researchers that we work with where they're investigating this problem in Ontario. But it has since spread fairly far and is causing a lot of damage to the beech, whether it's killing out trees in the canopy or even killing new developing trees in those infested locations. So like I had mentioned, the main symptom we're seeing is this veinal discoloration. It's kind of a mottled appearance at first, but over time it kind of will lead to almost I call it rugosity. That is the term when it leads to rough, textured tissue. And so if you put your hand on this, the beech leaf itself will be mostly smooth in comparison to the infected areas, which are going to be very rough, almost upraised as well, and discolored. The pathogen, which is a nematode, and I'll show you that in a little bit, infests the foliage, and eventually, as time goes on, it will infest the buds. So it will feed on newly developing leaf tissue in the buds, and they will then create damage for the next season. As this continues to go on, it's killing and damaging the leaves. The vigor of the tree will be reduced, and over time, it will stop producing as many leaves. It'll thin out and slow down and potentially even die. So, this is fairly problematic as it has been killing out mature beech trees in our native woodlands. Here's another image of the leaves. And so at first it starts off as yellow, but eventually it will start to turn, I'm sorry, it starts off as green and then it will turn yellow over time. We don't necessarily see much of this kind of necrotic area right here. That's something else altogether. But since you see this kind of band develop, you can see how it is that upraised portion, and it's going to be rough in comparison to the rest of the tissue. If infection happens at the bud, pretty much the entire leaf might develop this coloration and irregularity in form as it develops. And so on the right-hand side, you can see a lot of the affected leaves there on individual stems, which are colonized by the nematode. On the left-hand side, you can see a tree that's affected, that's losing, it's thinning out and it's losing its leaves. And like you can see here, it pretty much is an open canopy and eventually will die. Like I had mentioned, this is actually caused by a nematode. So nematodes normally are stuff in the ground. They're in the soil. And there's not that many plant pathogenic nematodes out there, but those that exist that we end up seeing on here, they're actually fairly devastating under the right conditions. Since our trees are highly susceptible to this problem, it just decimates them. But it is infecting the leaves and infecting the buds, like I had mentioned. This is called Lidolyncus, and it is thought to be native to Asia, or at least there are relatives in Asia that are very similar to the species. But here you can see them kind of wiggling in water, and they need free water to be able to move around. And so if there is not no water on the exterior of the plant and they kind of exit the leaf, they'll dry out and eventually die. But here on the slide, they're kind of just swimming around and they're aggregating in an area where there's some light. There are some problems out there that are lookalikes. So this is actually a problem that's caused by aphids. Now it looks very similar with the kind of vein, the banding between the veins, but it will curl. And often when you look on the underside, you try to unfurl that a little bit, you will see the evidence of the aphids over here on the underside of the leaf. We don't normally see a lot of curling with BLD or beech leaf disease. We just see that kind of upraised area and kind of that roughness. But if there's curling involved, or if it seems on maybe on one side of the leaf and it's not kind of sporadic and patchy, it's more clustered, it might be related to these aphids. If, however, you suspect that there are symptoms of BLD, of course, please send it into a lab and they can help verify whether or not pathogens there or not. Here is another example of something that looks similar. These are araneums. So they're araneum mites that will kind of feed on the tissue, lead to this irregular growth, and they are kind of bumpy. And so this is also kind of rough, but it produces a cluster of little, almost like glands together that is a little patch on both the upper side or even the underside of the leaves. These mites will be located directly in those little spots as well, but you can see that this is going to be a different texture. It's going to be lightly colored, and it's not solely within the veins of the leaf. So you can use that to slightly differentiate that from BLD as well. So managing this problem, there are, and I say fungicides here, but it's pesticides in general. There's actually a fungicide that has activity on nematodes. Nematodes are not fungi, they're actually animals. But it is a fungicide that has been known to have efficacy against nematodes, and they're evaluating that for use for beech leaf disease. But at this time, there are few to no products that are available for management of this pest. And likely not many that you can use widespread in a forested situation. So there has to be evaluation of that as well. Is it efficacious in a forest setting? Does it make sense to use that as well in terms of cost effectiveness as well? So there are products maybe in the future, but at the moment, just monitor the tree where possible. Over time, you might have to potentially chop the tree down and destroy it to try and save some of the trees around it. But we've been recommending people just monitor trees right now as the nematode spreads and causes damage. Now, how does it spread around? I didn't mention that earlier on, but it's not entirely certain. There's still research out there in terms of how it's spreading. We know it can move around in water, but that's not going to be moving from lake next to Ohio all the way to the East Coast, there's actually a thought that birds are eating the buds and then traveling and then defecating out those buds after they've eaten them and spreads that way. So that is one possibility, though. I mean, they're still not 100% certain how it's moving around. So, as it pertains to all of these invasive problems, including invasive insect issues that we didn't necessarily discuss today, please consider reaching out, whether to eddmaps.org. This is a organization that's in association with the Southern IPM Center, or IPM centers through the U.S. They will share that information to appropriate experts at universities as well as state departments of ag and so on, about possible reports of invasives. You can also, for Indiana, call and or reach out through the Report Invasive website, email our lab or even IDNR to try and let them know what you're finding and what you think might be there. When you report, please write down the location, GPS coordinates if you have that. Take a photo of the issue you're seeing and maybe even take a sample, but you don't necessarily need to take a sample if somebody says they're going to be coming to that location and check it out themselves. So please give us the information that you have and just be on the lookout. If you have any questions concerning these issues, feel free to reach out. We can tell you how to collect an appropriate sample, and maybe even what to look for if you're concerned about maybe boxwood blight and so on. But of course, when it comes to firewood, please don't move firewood around. Please only buy where you're going to burn it itself. So been a fantastic campaign, and I even got a free hat out of it. Don'tmovefirewood.org is a great place to go for that kind of information. So with that, I'll answer any questions. Thank you for your time.
All right, thank you, John. That was very, very informative. We do have one question in the chat. Can pruning help with beech leaf disease?
I mean, I don't know if there's a lot of information out there on it at the moment. There's only a few researchers evaluating management recommendations. But as far as pruning goes, on an individual plant, you could potentially remove the infected material. But there's also situations where we call asymptomatic infection. So that means that we have leaves that could be infected or infested by this nematode. But they don't necessarily cause any symptoms. There's not enough damage. Maybe they weren't in the leaf bud at the time to lead to that rugosity or that misshapen appearance to the leaf. So it's not 100% clear if that'll help very well because of these asymptomatic situations. I will say on top of that, and I'm actually a part of a survey that's being conducted with amongst 3 states. And in our conversations, this has come up from a few researchers where you can have significant disease symptoms one year, and then the next year you don't have any symptoms. Now, the nematodes aren't gone, but it's been difficult to find them. And this could be due to environmental stress, both stress to the tree or stress to the nematode itself, and it's not surviving as well. Or maybe some other impacts, but we don't have 100% continuation of symptoms from year to year. And depending on the scenario, those symptoms may abate a little bit, but then get worse a different year.
Okay, do we have any other questions for John? So David is asking, does he need to send in a sample to verify a potential laurel wilt infestation?
Yes, pretty much with any of these, we would recommend submitting a sample in. While there are a number of different things out there that can look very similar to the problems we've discussed. I put some lookalikes in there for beech leaf disease, but we have verticillium wilt, which can infect some of the same hosts of laurel wilt and cause very similar symptoms. Normally, this might be in the absence of a beetle, or beetle infestation, so that might give us an idea of maybe it's not laurel wilt, but ultimately we want to verify both by culturing out the fungus, maybe even using molecular tests, and then identifying the beetle with laurel wilt in particular, to ensure that we have the disease present. If we end up having just a visual, like photos, that kind of representation, we can't necessarily verify it's there, and they won't necessarily maybe put in any regulatory action, whether it's eradication of plant material, monitoring efforts, and things like that, until we get more data.
And always remember that Indiana and several other states do have extension offices throughout their states, and those extension educators can help you get started with those processes.
Very true. Thank you for bringing that up.
Let's see. And we have a question. How is laurel wilt moving?
So since it is a vector disease, it's moving around on the beetles themselves. So the beetles will fly from one location to the next and then attack a susceptible host. Kind of like with TCD, it's moving around by the insects. I will say though that um, they've been able to find that there are other beetles that can transmit the fungus, Rafflesia loricola. And so it's not necessarily just the red bay ambrosia beetle that we're worried about. So there are other native species that might be able to transmit the fungus as well. So this kind of goes back to the don't move firewood as well, since it's colonizing the wood and then the pathogen is inside the wood. You move that wood to another location, the beetles are going to come out and then they can spread the fungus to plants that are in that area. So once again, don't move firewood.
Okay, do we have any other questions? I am not seeing any pop up. So, what I'll say then is if you have additional questions, feel free to send them to me. I am happy to get them to John if you have them. Other than that, John, thank you very, very much for presenting to us today. That was a lot of great information. And hopefully, we will see everybody at our next session of Emerald Ash Borer University. I believe our next one is going to be covering Alum Zigzag Sawfly. So, everybody, have a great week, have a great Thanksgiving, and we'll see you at the next one.